Hello, welcome to Biology, the scientific study of life, DNA technology and genomics. Understanding and manipulating genomes. One of the great achievements of modern science has been the sequencing of the human genome, which was largely completed by 23. The sequencing of the first complete genome, that of a bacterium, had been carried out a mere 8 years previously. During the intervening years, researchers accelerated the pace of DNA sequencing while working on other genomes, aided by the development of faster and faster sequencing machines. These sequencing accomplishments have all depended on advances in DNA technology, starting with the invention of methods for making recombinant DNA. This is DNA in which nucleotide sequences from two different sources, often different species, are combined in vitro into the same DNA molecule. The methods for making recombinant DNA are central to genetic engineering, the direct manipulating of genes for particle purposes. Applications of genetic engineering include the manufacture of hundreds of protein products such as hormones and blood clotting factors. Using DNA technology, scientists can make recombinant DNA and then introduce it into cultured cells that replicate the DNA and expresses its genes, yielding a desired protein. DNA technology has launched a revolution in the area of biotechnology, in the manipulation of organisms or their components to make useful products. Practices that go back centuries are forms of biotechnology. For example, the use of microbes to make wine and cheese, and the selective breeding of livestock, which exploits natural occurring mutations and genetic recombination. Modern biotechnology based on the manipulation of DNA in vitro differs from earlier practices by enabling scientists to modify specific genes and move them between organisms, is distinct as bacteria, plants, and animals. DNA technology is now applied in areas ranging from agriculture to criminal law. More important, its use allows researchers in virtually all fields of biology to tackle age-old questions in a more comprehensive way. For instance, the level of expression of thousands of different genes can now be measured at the same time, as shown in the DNA microarray in this figure. Notice that. In the photograph, the color of each spot represents the relative expression of one of 2,400 human genes in a particular tissue. With this technique, researchers can compare gene expression in particular tissues or under different conditions. The knowledge gained from such global expression studies was largely inaccessible only a few decades ago. Okay, in this chapter we first describe the main technique for manipulating DNA, and then discuss how genomes are analyzed and compared at the DNA level. In the last section, we survey the practical application of DNA technology, concluding the chapter by considering some of the social and ethical issues that arise as DNA technology becomes more pervasive in our lives. DNA cloning permits production of multiple copies of a specific gene or other DNA segments. The molecular biologists studying a particular gene faces a challenge. Naturally occurring DNA molecules are very long, and a single molecule usually carries many genes. Moreover, gene may occupy only a small proportion of the chromosomal DNA, the rest being non-coding nucleotide sequences. A single human gene, for example, might constitute only 1 to 100,000 of a chromosomal DNA molecule. As a further complication, the distinctions between a gene and the surrounding DNA are subtle, consisting only of differences nucleotide sequences. To work directly with specific genes, scientists have developed methods for preparing well-defined gene sizes, species, or size pieces of DNA in multiple identical copies, a process called gene cloning. DNA cloning and its applications are preview. Most methods for cloning pieces of DNA in the laboratory share certain general features. Uncommon approach uses bacteria, most often Escherichia coli, and their plasmids. Recall that bacteria plasmids are relatively small from chapter 18. Circular DNA molecules that replicate separated from a bacterial chromosome. For cloning genes or other pieces of DNA in the laboratory, a plasmid is first isolated from a bacterial cell, and then the foreign DNA is inserted into it. The resulting plasmid is now a recombinant DNA molecule combining DNA from two sources. The plasmid is returned to a bacterial cell, producing a recombinant bacterium, which reduces to form a clone of identical cells because the dividing bacteria replicate the recombinant plasmid and pass it on to their descendants. The foreign gene is cloned at the same time, that is, the clone of cells contains multiple copies of the gene. Cloned genes are useful for two basic purposes: to make many copies of a particular gene and to produce a protein product. Researchers can isolate copies of a cloned gene from bacteria for use in basic research, or to endow an organism with a new metabolic capability, such as pest resistance. For example, a resistance gene present in one crop species might be cloned and transferred into plants of another species. Alternatively, a protein with medical uses, such as a human growth hormone, can be harvested in large quantities from bacterial cultures carrying the cloned gene for the protein. Notice, most protein coding genes exist in only one copy per genome, something on the order of 1 part per million of DNA. So the ability to clone such rare DNA fragment is extremely valuable. Back to overview of gene cloning with a bacterial plasmid, showing first uses of cloned genes. Notice that bacterium, bacterial chromosome, and this one, a gene inserted into plasmid and bacterial chromosome. Notice again here, notice that cell containing gene of interest, DNA chromosome, and this one, gene of interest. Notice that recombinant DNA plasmid put into bacterial cell, and recombinant bacterium. Gene of interest, gene copies of gene. Notice that host cell grown in cultures to form a colon of cells containing the cloned gene of interest. Protein expressed by gene of interest. Notice that protein harvested Basic research and various applications, not basic research to protein, and this one basic research on gene for past resistance inserted into plants, gene used to alter bacteria for cleaning up toxic waste, and this one protein dissolves blood clots in heart attack therapy, and human growth hormone treats stunted growth. These are basic research on protein, and this one on gene. Okay. In this simplified diagram of gene cloning in the laboratory, we start with a plasmid isolated from a bacterial cell and a gene interest from another organism. Only one copy of the plasmid and one copy of the gene of interest are shown at the top of the figure, but the starting materials would include many copies of each. Thanks for listening.